All right, well, welcome everybody to this week's edition of the EV Journal Club. Thank you all for joining. I made a little joke in our flyer for this week that we're not going to have pizza, we're going to have a Philly cheesesteak. And so it's my honor to introduce the Mỹ Mahoney lab, and May herself is here on the call with two of her students. So Philadelphia, as we were just discussing a few minutes ago, has really developed over the last few years into a center of strength in EV research with the multiple institutions there and a lot of good researchers who have EV-related research programs. So today we're going to hear about a recent publication in the Journal of Extracellular Vesicles that addresses squamous cell carcinoma and how EVs are playing into the development of this cancer along with their cargo, so their RNA and their protein cargo and associates. May, thank you so much for joining us, and perhaps you'd like to also introduce the members of your team, Bree and Joe, who are here.
All right, well, first, I want to say thank you very much for this very kind invitation. I look forward to sharing with you the work that went into our recent publication. I actually like these Zoom seminars because they allow me the opportunity to let my students who actually did all the work to present their findings. And with me today, I have Joe Flemming. Joe, can you wave? Yeah, he's a second-year master's student, and Brianna Hill, who is a second-year PhD student here at Jefferson. And I gave a shout out to them at the beginning, but before the audience came on, but I want to say that this work was made possible because of their vigilance and their devotion to this work, because they came in during the pandemic to finish up all the reviewers' comments and suggestions. So I will start the seminar, but I'm going to go ahead and stop the video of myself because I don't want to be distracted by my face. So here we go. All right. So next, Bree. Our lab has long been interested in the molecular mechanisms that drive malignant transformation of epithelial keratinocytes. And today we will focus on squamous cell carcinoma, cancers that arise from stratified epithelia of the skin, oral mucosa, esophagus, and cervix, to name a few. Now, there are more skin cancers than all other cancers combined. And SCC is the second most common form of skin cancer arising from chronic UV exposure. Cutaneous SCC is usually non-cancerous and most can easily be excised. However, a small percentage of cutaneous SCCs do metastasize and are extremely malignant. A significantly higher percentage of head and neck form of SCC or oral SCC is malignant. It is the sixth most common cancer with high rates of metastasis, morbidity, and mortality. Oral cancer is one of the top 10 most common causes of death. The leading cause of head and neck SCC is tobacco and alcohol abuse. Next, the ability of tumor cells to proliferate and metastasize is mediated by the mutual dialogue between those cells and their tumor microenvironment. Fibroblasts are an integral part of the tumor stroma and do actively interact with cancer cells to promote their initiation and progression. But how do epithelial and stroma cells communicate? It is well established that multicellular organisms are organized and maintained by complex systems of intercellular communication. And these include the cell-to-cell contacts through which adjacent cells can communicate through signaling. Cells can also secrete soluble factors such as cytokines, which then can recruit and activate cells of the immune system. Emerging as critical for intercellular communication are the extracellular vesicles. And because most of you guys are leading experts in vesicles, I will not go into any detail characterizing them. I just want to mention that in living cells, microvesicles or shed vesicles are derived from outward budding of the plasma membrane and their content often reflect the content of the cells. On the other hand, exosomes are generated from invagination of the membrane to form endosomes. These endosomes are destined for lysosomal degradation, or alternatively, undergo a second round of membrane invagination to form multivesicular bodies. These multivesicular bodies then fuse with the plasma membrane, releasing exosomes. These EVs are taken up by recipient cells through various mechanisms, including membrane fusion, receptor-mediated internalization, or endocytosis. Now, originally thought to function exclusively to remove trash or unwanted material from the cells, EVs have emerged to play critical roles in many biological processes, from controlling proliferation and differentiation to promoting angiogenesis. They are important regulators of cancer development and progression, as EVs are secreted in all bodily fluids, including blood and urine. They can be utilized as biomarkers. And more importantly, because they contribute to disease pathology, they can be targeted for therapy. It is well established that cancer cells secrete more EVs than their non-malignant counterparts. This necessitates a better understanding of the mechanisms that regulate EV biogenesis and content. During malignant transformation, cell-to-cell adhesion is often deregulated. And because adhesion proteins play a critical role in regulating membrane dynamics, we were interested in whether these proteins can modulate EV biogenesis. Our lab has long been interested in the desmosomal junction that provide tensile strength to cells and tissues by linking the transmembrane adhesive components to the intermediate cytokeratin filaments between the cells, as shown here by EM. Schematically, the transmembrane components of the desmosomes are composed of the cadherins, desmogleins, and desmocolons. Outside of the cells, these cadherins interact with in a heterophilic manner through ectodomain 1, as shown here. Inside the cells, they interact with the armadillo family of signaling proteins and the plaquin family of linkoproteins. These plaquins then bind to the intermediate cytokeratin filaments to provide cell-to-cell linkage and stability. In human, there are 4 desmogleins, and desmoglein 2 is the most ubiquitously expressed. Desmoglein 2 is highly expressed in epithelial and cardiac cells. In mice, loss of desmoglein 2 leads to embryonic lethality, resulting from defects in the embryonic stem cell growth and development in the inner cell mass of the blastocyst without affecting cell-to-cell adhesion. This was the first evidence that desmoglein 2 plays a role outside of adhesion. Now, in human, mutations in desmoglein 2 is linked to cardiomyopathy, resulting from loss of myocytes, an extensive increase in fibrosis, and adipocyte infiltration. And interestingly, in human pluripotent stem cells, desmoglein 2 is critical for self-renewal and formation of embryonic body and teratomas, which are tumors of germ cells. Desmoglein 2 also mediates the epithelial to mesenchymal transition through a beta-catenin and slug pathway. Now, contrary to the general consensus, the cell-to-cell adhesion is downregulated in cancer, particularly during metastasis. Desmoglein 2 is highly upregulated in many cancers. In fact, 17 of the top 20 most common cancers express desmoglein 2, including skin cancer, shown here. However, desmoglein 2 is highly upregulated in many cancers, including BCC and SCCs. And furthermore, SCCs and BCCs are the two most common forms of skin cancer. Furthermore, desmoglein 2 is also upregulated in melanoma and correlates with poor prognosis. Next. In normal oral mucosa, again, desmoglein 2 is expressed at low levels in the proliferative basal cell layer, but highly upregulated at the leading edge of oral SCCs. And most importantly, desmoglein 2 level correlates with poor prognosis, as shown here in the right. Now, to mimic BCCs and SCCs, we generated two transgenic mouse models overexpressing desmoglein 2 in the basal cell layer under the K14 promoter and in the superficial epidermis under the involucrin promoter. And with the involucrin transgenic, we immediately observed extensive hypoplasia, susceptibility to chemical-induced carcinogenesis, as well as spontaneous tumors. However, the K14 promoter showed no change in epidermal phenotype, as you can see here, but further analysis showed enhanced wound healing with evidence of desmoglein 2 activated at the leading edge of the wounds. These results further support the emerging concept that cancer exploits the normal wound healing process to promote deregulated growth and metastasis. So with this background and the fact that EVs play key roles in tumorigenesis, we wanted to know whether changes in membrane presentation of desmoglein 2 would affect vesicle biogenesis. And furthermore, do these secreted vesicles contribute to tumor growth and progression? So in 2017, we published a paper in FACEP where we showed that desmoglein 2 does modulate EV biogenesis and pathogenic content. And furthermore, desmoglein 2 EVs can enhance stromal fibroblast growth and activation, which brings us to this recent publication in JEV. Here, we further studied how intercellular communication by the cell-to-cell adhesion through desmoglein 2, cytokine release through IL-8, and EV biogenesis and uptake, and these three means of intercellular communication is well coordinated. And Joe, take it away.
All right, so to start off, our method of EV isolation from conditioned medium is indicated on the left of this slide. Briefly, cells were removed by spinning at 300 G for 10 minutes, followed by the removal of apoptotic bodies with a 2000 G spin for 10 minutes. Then large EVs were pelleted with a 30-minute spin at 16,000 G, and small EVs were pelleted with a 70-minute spin at 120,000 G. So next, the EVs we isolated were characterized via nanoparticle tracking analysis, indicating a larger population of EVs above 200 nanometers in the top profile found in our 16K pellet, and the characteristic small EV profile below 200 nanometers in the bottom profile found in our 120,000 pellet. Small EVs were imaged via transmission electron microscopy, indicating that successful isolation of intact vesicles from our procedure. Next, on the right by Western blot, our isolates showed enrichment of the small EV-associated markers CD63, CD9, and flotilin-1, with an absence of the mitochondrial marker COX-4, indicating no cellular contaminants. So next, it is known that many membrane proteins, including desmoglein 2, undergo protein palmitylation, which is the reversible addition of palmitate, the 16-carbon fatty acid to amino acid residues, which is illustrated in the cartoon found at the top right, and plays an important role in the proper function of many proteins in capacities such as protein-protein interactions and proper membrane trafficking. Addition is catalyzed by membrane-bound protein acyltransferases, and removal is catalyzed by cytosolic acyl protein thioesterases. So next, we designed an experiment to address whether this modification is important for small EV biogenesis. To accomplish this, we used a global inhibitor of palmitylation and 2-bromo palmitate is a broad inhibitor of proteasyl transferases, indicated in red on the cartoon. And as you can see here in the bottom right with these two SCC cell lines, desmoglein 2 enhances EV release, but 2-bromopalmitate abrogates this effect. However, since 2-bromopalmitate inhibits palmitylation on a global level throughout the cell, we needed to address whether these results were due to desmoglein 2 dependent mechanisms. We next made mutations in two of desmoglein 2's cysteine residues adjacent to the transmembrane domain, shown here in yellow in the top left, which we have previously shown to be responsible for all palmitylation of desmoglein 2. And by site-directed mutagenesis, we changed the amino acids to alanine or serine and generated these constructs. We used these to establish SCC cell lines expressing a mutant desmoglein 2 unable to be palmitylated. So using our established cell lines, we tested the effect mutant desmoglein 2 has on EV biogenesis. And by Western blotting in the top right, it is clear to see that all three cell lines express similar levels of endogenous desmoglein 2, while the modified cell lines express similarly high levels of the exogenous GFP fusion proteins. So next, through IF in the bottom left, a strong GFP signal is noted throughout the cytoplasm of the control cells, while the desmoglein 2 overexpressing cells demonstrate the typical cell surface presentation of desmosomal proteins. This is as opposed to the mutant desmoglein 2, which shows much stronger cytoplasmic staining of the GFP fusion protein, indicating inefficient trafficking to the plasma membrane. Next, we collected conditioned medium from the cell lines and purified EVs by ultracentrifugation and analyzed by nanoparticle tracking analysis. In the bottom right, we see desmoglein 2 enhanced EV release, while the loss of desmoglein 2 specific palmitylation abrogates this effect. So therefore, we concluded that palmitylation of desmoglein 2 is necessary for its function in EV biogenesis. So next, to again confirm the altered EV biogenesis results, we performed a bottom-loaded density gradient separation on EVs from our three cell lines. So next, as you can see on the right, by CD63 positive fractions found at the interface of the 5 and 30% fractions, desmoglein 2 overexpressing cells contained a much stronger signal, indicating more EVs. In the mutant cells on the bottom, there's a large right shift in CD63 signal into the 30 and 40% fractions, indicating an alteration of EV structure. A recent publication has indicated that subpopulations of low and high density EVs exist and may have distinct molecular cargoes. And we're currently investigating the difference between the cargo of these high density vesicles, and we're excited to see those results sometime soon. So next, in order to elucidate the mechanism by which desmoglein 2 alters small EV biogenesis, we performed immunofluorescence on our SCC cell lines. Starting with staining for desmoglein 2 in the top left, note the enhanced desmoglein 2 staining in red for the cell lines overexpressing desmoglein 2, and an abrogation of this cell surface staining in the mutant cells. We next sought to observe the effect desmoglein 2 has on endosomal markers beginning at the plasma membrane. Flotilin 1, shown below the desmoglein 2 staining, is a lipid raft protein important for EV biogenesis. Note the increased surface presentation with expression of desmoglein 2 and an ablation of this effect and expression with the expression of mutant desmoglein 2. In the bottom left and top right, respectively, we stain for EPS15 and EEA1, which are another set of early endosomal markers. The trend we have previously shown in the desmoglein 2 overexpressing cells and the mutant desmoglein expressing cells is conserved with these additional markers. These results show that desmoglein 2 modulates small EV biogenesis through alteration of markers early in the endocytic pathway. And in order to confirm that desmoglein 2 is altering the early plasma membrane adjacent elements of the endocytic pathway, we stain for RAB7, which is a GTPase that plays a role in the late endosomal fusion with a lysosome. These results indicated no change with expression of any of our constructs. But before moving on, it's worth mentioning that we have shown caveolin 1, which is a lipid raft protein known to inhibit EV uptake, is downregulated in response to desmoglein 2, but excluded this data because it has already been published. Next, by Western blotting here, we show that desmoglein 2 enhances the protein production level of our endosomal markers in addition to the altered subcellular localization shown in the previous slide. So next, in order to supplement our genetic perturbation of EV biogenesis, we implemented pharmacological means methods to confirm that desmoglein 2 enhances EV biogenesis through the endocytic pathway. The schematic to your left shows the inhibitors we chose, as well as the section of the endocytic pathway that they affect. Desmoglein 2 overexpressing cells, or GFP control cells, were treated with baflamycin A1, a proton pump inhibitor, which prevents acidification of endosomes, thereby blocking their eventual fusion of the lysosome, causing an increase in number of multivesicular bodies fusing with the plasma membrane. Also, they were treated with GW4869, which is a neutral sphingomyelinase inhibitor that inhibits the second round of invagination to form multivesicular bodies, therefore reducing the number of vesicles in these bodies and causing less fusion with the plasma membrane. Inhibiting these steps in the endocytic pathway was able to enhance or decrease the ability of desmoglein 2 to alter EV biogenesis. The figure to your right is a quantification of particles released into the conditioned medium by each of the SCC cell lines after treatment with these agents. In blue, desmoglein 2 is again shown to enhance EV release in our SCC cell lines. In red, GW4869 reduced desmoglein 2's ability to enhance EV release, and in yellow, baflamycin A1 greatly enhances EV release in both the GFP control as well as desmoglein 2 expressing cells. These results, in combination with our previous immunofluorescence data, confirmed that desmoglein 2's ability to enhance EV release is dependent on the endosomal pathway. So next, to assess the role EVs play in tumor development, we use a tumor xenograft model indicated in this schematic. 1 million cells were injected subcutaneously, and immunodeficient animals were monitored for up to 4 weeks unless tumor burden required an animal to be euthanized prior to the end of our experiment. So next, in the top figure, A431 SCC GFP control cells, shown here in black, provided us with a baseline for tumorigenicity of our SCC cells. When desmoglein 2 is overexpressed in these cells, shown in red, their tumorigenicity is greatly enhanced, and this alteration of tumor growth correlates with their enhancement of their EV release in vitro. Alternatively, when the SCC cells express our mutant desmoglein 2, shown in blue, their tumorigenicity is significantly reduced, again correlating with the ablation of their in vitro EV release. In order to further confirm the effect of our constructs on tumorigenesis, we generated cells using the head and neck SCC cell line UMSCC1, and similar results were observed, shown in the bottom figure. Next, through immunofluorescence of the excised tumor tissue, now in the middle, we see that GFP is localized in the cytoplasm of the control cells. In the GST2 overexpressing tissues, we see the normal pattern of desmosomal proteins found in the cell surface. This is as opposed to the mutant protein, which is expressed at a similarly high level, but is found at greater proportions in the cytoplasm. These results show that the altered function of our mutant desmoglein 2 is maintained in vivo. Next, most importantly, we isolated blood from animals in each of our tumor categories, and from that purified EVs. We found that mice having desmoglein 2 overexpressing tumors also had the highest number of circulating EVs, while animals that express mutant desmoglein 2 tumors had a significantly reduced amount. Through these results, we have shown that tumor burden correlates with total circulating EVs. So next, in order to address whether EVs are directly enhancing tumor growth, we repeated our tumor xenograft experiment, but this time we injected 20 micrograms of EVs from desmoglein 2 overexpressing cells, along with 1 million GFP control cells at day 0. The SCC GFP control cells are again shown in black and produce the smallest tumors. The desmoglein 2 overexpressing cells are shown in red, and the expression of our construct once again greatly enhances tumor development. The GFP cells plus small EVs, shown in blue, produce significantly larger tumors than the GFP cells alone. We saw that this one-time injection of EVs was able to increase tumor growth of control cells even multiple weeks post-injection. Now Bree will take over.
As discussed earlier, intercellular communication is comprised of complex systems that include cell-to-cell contact, cytokines, and EVs. It is well established that cytokines secreted by different cell types, including endothelial cells, macrophages, and fibroblasts, as well as increasing evidence of the secretion of cytokines by epithelial cells as well, can initiate signaling cascades in other cells. In order to assess if cytokines could be influencing signaling by associating with EVs, we utilized a cytokine array. We subjected conditioned medium and large and small EV fractions isolated by ultracentrifugation from cells overexpressing desmoglein 2 and parental controls to a cytokine array. The blue boxes demarcate the positive controls, and the black boxes surround the negative controls. In response to desmoglein 2, a number of cytokines were increased in the conditioned medium and small EV fractions. No major changes were observed in the large EVs. The red boxes show IL-8. These results were quantitated using densitometry on ImageJ software. A secretion of 8 cytokines were upregulated, while 2 were downregulated in the conditioned medium. However, in small EVs, 11 cytokines were enhanced in response to desmoglein 2. We found that desmoglein 2 overexpression also increased the levels of the pro-inflammatory cytokines IL-8 and IL-6, with IL-8 being enriched in both the conditioned medium and the small EV fraction, while IL-6 was only enriched in the conditioned medium, suggesting preferential loading of cytokines into small EVs. This data is summarized in these 2 charts. The one on the left is from the conditioned medium fraction, and on the right are the cytokines whose levels were altered in the small EV fraction. You can see that desmoglein 2 expression induced the production and packaging of molecules of different classes, such as the chemoattractants IL-6 and IL-8, as well as the adhesion molecule, an adhesion molecule involved in metastasis, ICAM1, and growth factors. The classic model of cytokine signaling is defined by the thought that cytokines that are secreted from cells or expressed on their surface then exert their effect on other cells and other cell types, including macrophages and other tumor cells. This is established as the case for IL-8 during tumor development. But if cytokines are released, they have to be available to activate their receptors. And if they were encapsulated in EVs, they would not be able to exert their effect. Fitzgerald et al. Recently published a beautiful paper showing that cytokines can be associated with the surface of EVs, thus maintaining their signaling potential while in the EV-bound state. Seeing an increase in the protein level of IL-8 in EVs, we next were interested in figuring out if IL-8 was on the surface of the vesicles, which could help it maintain its signaling capability. In order to do this, we isolated EVs through ultracentrifugation and floated them over an iodixinol gradient. And then, using the CD63 enriched fraction, we subjected it again to cytokine array and confirmed the presence of IL-8. We then subjected this SEV fraction to gentle trypsin digestion. The rationale being if the IL-8 was protected from degradation because it was inside of EVs, then it would not be degraded. However, if the IL-8 was on the surface of the EVs, it would be degraded. And as you can see on the right, when subjected to trypsin digestion, IL-8 was in fact degraded, suggesting that IL-8 is found on the surface of EVs. Upon seeing that desmoglein 2 modulates IL-8 levels, we wanted to delve deeper into the regulatory network that was being altered in order to increase IL-8 expression in these cells. We know that microRNAs can be transported in EVs and regulate protein expression post-transcriptionally. MicroRNAs are first transcribed in the nucleus, processed by Drosha and then by Dicer, and loaded into the multivesicular bodies and secreted in EVs. This led us to analyze the microRNA content of cells and EVs in response to desmoglein 2 overexpression. Through RNA-seq, we found that 93 microRNAs were altered in cells, and 19 were altered in EVs. Only 8 of these were shared, suggesting active sorting of microRNA content into EVs. It was found that the most dramatically depleted microRNA in both cells and EVs in response to desmoglein 2 overexpression was miR146A, which does target IL-8 indirectly. It's important to note that miR146A is a negative regulator of myeloid cell proliferation and is also downregulated in breast cancer. Next, we need to determine if the downregulation of miR146A was sufficient to increase IL-8 expression. We know that when IRAK1 is activated, it induces NF-kappa B's translocation to the nucleus and induces transcription of target genes, including IL-6 and IL-8, as well as miR146A and B. miR-146A then functions as a negative feedback mechanism for this pathway and reduces IRAK1 expression. In order to show that miR-146A modulates IRAK1 and therefore also IL-8 expression, we treated SCC cells with miR-146A mimic, an inhibitor, and a scrambled microRNA, and then using qPCR, assessed the amount of IL-8 or IRAK1 expression. We found that treatment with the miR-146A mimic decreased the amount of IL-8 and IRAK1, while the miR-146A inhibitor increased the amount of IL-8 and IRAK1 in the cells, showing that inhibition of miR-146A was sufficient to alter IL-8 levels in vitro.
Okay, so finally, to establish the clinical relevance of our in vitro and in vivo findings, we wanted to know if there's a correlation between desmoglein-2 and IL-8 expression in the tumors. We obtained 14 head and neck SCC tumors from surgery and performed RNA-seq. The graph to the left is a correlation analysis between the expression of IL-8 and desmoglein-2 in these tumors. It demonstrates a moderate linear correlation with a Pearson R-square coefficient of 0.52 and is statistically significant. This is quite promising considering the whole heterogeneity of the whole tumor tissue. This is quite, it also suggests that perhaps with better analysis such as nanostring, we may get a stronger correlation between the two proteins. Finally, to the right, to determine whether circulating plasma level of IL-8 could be a predictive indicator of response to treatment, we collected blood plasma from 28 head and neck patients prior to treatment with PD-1 inhibitor nivolumab and the vasodilator tadalafil. IL-8 level was measured by ELISA. Subsequent to drug treatment, patients were stratified according to clinical response, showing those who responded to the drug treatment had significantly lower IL-8 level as compared to those that did not show a positive response. These results suggest that perhaps there is a need for combinatory treatment to lower IL-8 level prior to or in combination with immune checkpoint therapy. So next, in this schematic diagram, we propose that because desmoglein-2 promotes cell growth and survival, there is a selection for cells that overexpress desmoglein-2, which then enhances EV release. Desmoglein-2 also downregulates miR-146A. The mechanism is currently not known. We do know that loss of miR-146A abrogates the inhibition on IL-8 expression, and the combination of small EVs and IL-8 can promote cell proliferation, migration, and tumor development. Now, in summary, I hope our work has demonstrated that intercellular communication through cell-to-cell adhesion, cytokine release, and secretion of EVs is well coordinated, and it is critical for tumor growth and development. And most importantly, the subset of head and neck patients that do not respond to immune checkpoint therapy may benefit from treatment targeting IL-8. And with that, I would like to thank the members of my lab who contributed to this work, particularly Joe Flemming and Brianna Hill, who you have heard from, as well as Wasaif Hogg, who just left us for dental school. My collaborators, Jim Wall from the University of Nebraska for the cell lines, Kenneth Tsai from the Moffitt Institute for the RNA-seq analysis, the microRNA-seq analysis, and Peter Vermert for the EV analysis, and Claudine Bonder in Australia for the work with the cytokines. Most importantly, and I have to say this is really important, the SCC research group to the right there is organized by the Department of Otolaryngology, Head and Neck Surgery at Jefferson, without these people, this work would not be possible. So finally, to thank my funding agencies, the first and most important one is the Society for Investigative Dermatology, that have supported me throughout my entire career, and research funding from the National Institutes of Health through NIAMS. And we made this seminar a little short so that you will have time to ask us any questions.
Wonderful. Well, thank you so much, May, for presenting this work, and also to Bree and Joe for their very clear presentations. And I'd just like to ask that everybody please propose your questions in the comment box. I see that we've already have some that have come in. So we will go ahead, and I'm going to allow people to unmute themselves. And I'm going to just go through the list. It looks like we have, we'll start with somebody local. So Lucia Languino.
Thank you, Ken. Great, and really congratulations on organizing this journal club series. It's really enjoyable. Thank you, Ken. Okay, so May, Joe and Brianna, super, congratulations. Okay, so the picture you showed for desmoglein GFP, it shows a cortical area, cortical actin area. Is that what it's supposed to be? Because right, the way you presented at the beginning, there are specific structure where it's supposed to be localized.
Right. So that's a really good question, Lucia, and I know where you're coming from with that. I think that it's sometimes very difficult to get really good immunostaining, and depending upon the image and how you obtained it, it can show punctate cell-to-cell borders, or it can show other kind of hazy, staining. So, however, having said all of that, there is strong evidence that desmoglein 2, under certain pathological conditions, desmoglein 2 can switch between associating with keratins and with actins. So and I appreciate you observe that, because I would love to talk with you more on where do you see that cortical actin staining, and that would be really nice for us to actually have a little group meeting between your group and our group to educate our students on how they can detect the two staining. So yeah, no, I would love to know what role it plays, because I think association with cortical actins play a very important role in EV biogenesis and angiogenesis and all that from your work. And then from our work, we are seeing that this very similar type of function. So whether or not it's associated with a desmosome, or is it associated with the actins, we need to really work on that together. So that's a good collaboration project right there.
Thank you. That deal. Okay.
Sorry, I just got a message that the host wants me to unmute myself. So I thought I was the host. Anyway,
we didn't do anything.
I sometimes have some good conversations with myself, as you can imagine. Anyway, let's move on then to Paula, who asks about distinguishing between different kinds of tumors. Paula?
Yeah, I was just wondering, in the patient samples, if you distinguish between the HPV positive and negative populations, since HPV positive patients typically respond better than the negatives.
That is a great question, and we did not have a large enough population to do that, but we are doing that right now. So in collaboration with a company called Clara Biotechnology, they use a new technology to isolate exosomes, and we sent them samples that could distinguish HPV positive and negative, response to treatment and not response to treatment, and we're hoping with that we would be able to better analyze the data. But when we did this experiment initially, we did not have enough samples and we did not have the purity of the samples to do that kind of analysis. So we opted not to pursue that in this paper. But I agree with you. That's a really great question, and we should be able to give you that answer very, very soon.
Wonderful. Great. That sounds good. Our next question is from Stephen Amoa. Stephen, go ahead.
Hi, really nice presentation. So I'm wondering, especially the therapeutic side towards the end of the presentation, you mentioned the anti-IL-8 therapy, and I'm wondering, given the reduced miR146 expression, I mean, with microRNAs being the dominant cargo in exosome, is there any ideas towards either boosting their production, either endogenously or maybe getting them exogenously? I mean, I don't know, but could that be something to be looked at? Thank you.
Absolutely. And I have to tell you that this, our work has really opened a huge can of worms, and the BMS, Bristol Myers Squibb, which has an antibody to IL-8 as well as nivolumab and tadalafil and all that, and we contacted them and said that we were interested in doing a first thing is the combination between anti-IL-8 and nivolumab, and we found out that that unfortunately somebody beat us to this, but the good thing is that the pandemic shut everything down, so we're still in the same boat. So hopefully we can do this combination. And then Joe is right now working on the miR146A, how to target miR146A mimic into vesicles and use that as a target, as a therapeutic means. So yes, we are working with BMS for the anti-IL-8 level, monoclonal antibody, and we ourselves are generating the vesicles that we can use in therapy. So, you know, I don't know how long this will take because I'm a basic scientist, so I can only hope that my head and neck research group can help me figure this all out and try to go down that pathway of pursuing dual targeting of miR146A as well as IL-8. Did that answer your question?
Absolutely. So
Okay.
be watching that space.
Yes, we're working on it. We're trying to.
All right, thanks for the question. Our next question is from EJ Kwan about the location of the IL-8 receptor.
Bree, do you remember if there was IL-8 receptor, the CXCR1 on the vesicles? Did we see a difference? I can't remember.
Yeah, just I have really like a general questions. I might miss some slides that I just sitting on my desk several times. So it's a really basic questions. Have you seen any like a cytokine receptor in EV membrane, especially have you done the mass spec of your cells? And do you think that it's like can you say it's like a specific for the cancer cells? It has like a cytokine receptor in EV membrane.
I honestly cannot remember whether or not the IL-8 receptor was on the array or not. So I'm going to have to look back.
I remember the CXCR1, I guess.
Yeah. Bree, do you see it at all? I can't remember if we saw that.
We did look at it, May, and it's not on that array.
Okay, so it wasn't, but we never pursued it by looking at QPC. Or no, we would have to do a Western of the EVs. No, we have not done that. I'm sorry, we should at least, that's a great question, and you guys should at least try that when you guys get a chance, okay?
Okay, thank you.
Mm-hmm.
Good suggestion.
Thank you.
Our next question is from Chris Aishin or Aishin. About microRNA. Yes, go ahead.
So interesting talk. The microRNA that you found, can you hear me?
Yes. Mm-hmm.
Yes, okay. The microRNA you found, the 146A,
Uh-huh.
on your list, you had a lot of other microRNA on there. Some were, I noticed, I was quick up there, so I was looking quick, but I missed some of them. Some were oncomeres, others had other functions. I guess I'm trying to figure out, why did you focus on just this one? And do you think it's just this one, or could other microRNAs be involved that contribute to the effects you see?
Okay, that is a really, really good question. And yes, we are pursuing this. The most important, the other microRNA that we're pursuing heavily is miR-155. And
Mm-hmm.
miR-155 and 146A kind of work in concert together. And they both work they target the NF-kappa B pathway, which is really is a really great pathway. I mean, it is a pathway that regulates IL-8 expression, as well as other pro-inflammatory cytokines such as IL-6 and ICAM-1. So with that, we are pursuing this path. I mean, we're pursuing several other microRNAs other than just miR-146A. We only, for a paper, you have to be really focused. And I did not want to start, talking about all the other microRNAs because it wasn't a microRNA paper. But in the grant, I promise you, the other microRNAs are, discussed, particularly the interaction or the dual play between miR-155 and 146A, how one is upregulated and the other one is downregulated, and which one is the master regulator, etc. So yes, we are doing that, and Bree, who loves big, systems biology, so she's looking at the different pathways that could be targeted by all of these microRNAs. So more is to come is all I can tell you, but we just wanted, for the paper, we wanted just to focus because we wanted to talk about IL-8, but of course there are other pro-inflammatory. As you noticed, there were 11 cytokines that were upregulated in response to desmoglein 2 in these EVs. So we have to pursue the other cytokines, and we also have to pursue the other microRNAs, which microRNA is targeting which cytokines. So we're working on that. Thank you for that question.
Yeah. Well, just you may already know this, but we do a lot of this kind of microRNA work. So if you need any help, just reach out. We're happy to help.
Thank you. Thank you so much. We really appreciate that. We definitely will. We definitely will go upstairs. So Ken, Chris is just right upstairs from us.
Oh, wonderful. Okay, I was about to say, be careful what you volunteer there. No, that's great. That's great that to hear that, and certainly it's good to, I think one of the positive things from this pandemic right now is that we are having some opportunities to get together in ways that we didn't before. So
Right.
that's very good. And it looks like we've gone through the questions that are in the chat box, and of course, if anybody has anything else, please bring it up. But I wanted to, maybe as we get to the end of the hour, May, I know that you're somebody who thinks a lot about how science works and inclusion in science and, what we can do better. And, you mentioned to me when we were discussing that you're relatively new to the EV field. Of course, I think a lot of people are relatively new to the EV field. It's grown very rapidly. But as somebody who has thought about this and, has made some nice contributions now in the EV field, what do you see as something that I think that maybe the EV field could learn or could do better or differently? Is there anything like that has come across to you, as you've interacted with different societies and groups?
That's a really great question, and I think that targeting non-cell entities like EVs is it's so important, and we have to look back at all the previous works by all the people who looked at gene regulation and how they're regulated by in a non-cell method like EVs. We have to look back at those papers and see what did they find, and how it would be, you know, how it can be related to what we work on. Because I know that, you know, in talking to a lot of scientists out there, when I present work on EVs, people say to me, oh yeah, I used to study this, like, you know, RNA polymerase 1 or 2, or et cetera. And I found that, you know, there was something in the cell, outside of the cell, that was regulating it, because I didn't know at that time about EVs. So I think that's the direction we need to figure out, is what's the content of these EVs and how they can go into a recipient cell and regulate, even down to, you know, DNA proliferation, DNA polymerase, well, now I'm like mincing my words, but you know what I mean? I think we have to really look back at some of the earlier research, and when a question could not be answered, we need to figure out if those could be answered by the content of the EVs that we see.
So I really like that comment. You know, it may be that we have something here that could explain some of those past questions that have remained unanswered.
Right.
So in that sense, it is wonderful to see how many people are coming into the field and contributing their expertise in many, many different areas. We might be able to come up with more ideas where we can apply our knowledge of EV biology. So.
And I also think, I mean, for me, I really like the connection between wound healing and cancer development, and how a cancer cell has hijacked this wound healing phenotypes in order to promote cancer development. So we need to know what are the key players in wound healing, and how they can be hijacked by a cancer cell to promote their proliferation and survival. So a simple molecule like desmoglein 2, which mediates, you know, MAP kinase signaling through EGFR and CSARC, can now be hijacked by a cancer cell to then, you know, permit the cells to survive in a non-adhesive environment. So this would promote metastasis, and how a cancer cell can then use the cytokines that are released by these, you know, downstream of desmoglein 2 to promote it. And the other question is, how are these cytokines bind or associate with these vesicles on the outside? That's what I'm really interested in. How, I mean, what is it binding to that allows it to be protected by the environment in the bloodstream or in the urine, right? And how are these cytokines, I mean, how are these EVs so resistant to degradation? That's really fascinating to me. So we need to figure, there's so many questions to be answered now. I mean, that needs to be answered through, you know, in this field.
So maybe these are a few topics that we could revisit in the future EV journal clubs. So thanks for the ideas there. And I think we're coming to the end of the hour, so we'll conclude for today. But thank you again, May, Bree, Joe. Thanks so much for your presentation, for sharing your time and your expertise with us. If anybody does have any ideas for future journal clubs, things that you or your lab mates, members would like to present, whether it's your own work or someone else's work, please just reach out to me, and we'd be happy to schedule you one of these weeks. So thank you again, everyone, and I look forward to seeing you again soon.